Hi guys, welcome to my YouTube channel Unique Way of Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, after again long time I am here with the next session called as mutation. Okay, so mutation, it comes under your fifth chapter, that is principles of inheritance and variation. As in the last video we have discussed about determination or sex determination. Okay, and now this mutation is in your NCERT textbook on page number 87. Okay, on page number 87 in your second year textbook you will find this. Now we shall discuss about that mutation, which has been given in hardly half of a page information, but very important mutation, because if you know how does the mutation occurs, what is actually mutation, then it is very easy to understand the further part of a chapter or the next chapter. Okay, anyhow, we shall begin with what is mutation. Some information is there in your textbook also, you can refer it. I have made a note as I used to do it earlier from the beginning, same I did here also. You can find I have prepared a note of mutation where it has been mentioned as alteration of DNA sequences, which results in the change in genotype and phenotype of an organism. Okay, this is what alteration or alternation is. What it is, alteration is what it is, nothing but change. Okay, change of DNA sequences. What does DNA consist of? DNA consists of nucleotides. Okay, with nucleotides, that DATP, DGTP, DCTP, DTTP. Hope you remember this. Okay, if DNA sequences have been altered, then what happens? It results in change in genotype. What was genotype? That is, the sequence of DNA will get changed. Okay, of a gene will get changed, and phenotype of an organism. What is phenotype? Phenotype is nothing but an external appearance, as you know this. Okay, if DNA sequence have been changed, then it will change the genes and even it will change the external appearance, okay, of an organism. This is what mutation. Mutation, you can write this definition, or you can write this also. Change in a DNA sequence is called as mutation. If DNA sequence have been changed, we call it as mutation. Okay, any one of the definition you can mention there. Now actually, how does it happen? Okay, DNA sequences, how they are arranged. If we draw DNA, something like this, okay. Now this is the DNA. Now this DNA consists of some base pairs or some sequences on it. If I draw it only one strand, something like this, I am drawing one strand, and this is the another strand, two strands. Here it has been coiled, here it is uncoiled. Okay, now what does this DNA consist of? It consists of information. What information? That is our physical appearance. It might be, it consists of lot of genes. Which gene is responsible for protein? Every changes, everything which occurs in the body, that is because of DNA. Okay, now we are saying DNA consists of information. In what way? It is having one language of codes. Okay, like as we know, nucleotides are four, that is adenine, guanine, cytosine and thymine. Is it? These are the four nucleotides which you will get in DNA. In the first year you might have studied about this. Okay, adenosine, guanosine, cytosine and thymine, right? These are the four. Now this itself will be arranged in a sequential form on a DNA. If I am writing A, T, G, C, okay, C, C, G, A, something, whatever sequence are there, these will be arranged in all permutations and combinations on DNA. This is one strand. What will be another strand? The another strand will be having complementary base pairs of this strand. So if there is a A, it always pairs with T. If there is a T, it pairs with A. G pairs with C, C pairs with G. Okay, again here C, G, C, G. Here G, it will be C. Here it is A, it will be T. Okay, these are the base pairs which will be always will be together, and they are complementary to each other. Now this complementation and all you have studied in your first year, and even in the next chapter you have that information. While discussing about that, I shall discuss in detail. Anyhow, this is the sequence. What we say, I have shown only few sequences. It will be as will along the length of the DNA is there, it will be having that information on the DNA strands. Okay, this is the information. If there is, this is the actual sequence of the DNA, just will imagine. Okay, this is the actual sequence of DNA. But here in mutation, what happens? There is a change in a DNA sequence. In place of this sequence, if it is having, I shall write this one, if it is having A, T, G, G, C, C, G, A. Can you find any difference in this sequence and this sequence? Yes, definitely. Here in place of C, it has been placement of G, right? So C has been replaced by G. Then it has what we'll say, it is a change in the DNA sequence. Now DNA sequence has been changed. Actually it should be like this, but it has been changed to this. So that we call it as mutation. Here I have shown only one has been changed. It can be many sequences might have been changed. If many sequences might have been changed, that also mutation. One has been changed, it is mutation. If DNA sequence has been changed or it has been altered, we call it as mutation. See here, if it is G, then what you will expect here in place of G? It must be C. If this gets changed, then definitely the other strand also gets changed. For that we call it as mutation. Okay, fine. Now hope you understood what is mutation. Definitions also have been given here. Now we shall see what are the causes of mutation are. Causes of mutation are like there are many chemical and physical factors. Okay, which means mutation is caused by chemical factors and physical factors. Not only chemical, physical factors are also involved in that. UV radiations can cause mutations, which means one of the physical factor is what? UV radiations. Ultraviolet radiations, they can cause mutations. Okay, if there is a mutation in our body, in our DNA, then we might be having some mutational disorders. Okay, we might be having some diseases because of change in the DNA sequence. So UV radiations can cause mutation. Mutations may be caused due to errors in DNA. Okay, errors is what? Mistake. Okay, if this is what mistake now. In place of C, if it was G, then that is a mistake. When this mistake takes place? During duplication. That is when DNA will multiply itself, when DNA will replicate. Okay, replication of DNA we have in next chapter. But what I am talking about is duplication and replication. You might have studied about cell cycle, where you can find synthesis phase, S phase in cell cycle. Hope you remember. There are three major phases, that is G1, S and G2. Okay, and rest phase is also there. So then during S phase, what happens? Synthesis. What is synthesizing? DNA. So because DNA, while synthesizing itself, while replicating itself, it can undergo some mistakes, some errors, which can lead to mutation. Okay, so mutations may be caused by chemical and physical factors. UV radiation is also one of the major factors because of which mutation can occur. Mutations are caused due to errors in DNA duplication or replication. Duplication or replication both are the same, where one DNA is going to replicate itself into two DNAs. Okay, one DNA will replicate into two DNAs. Okay, one more is there, mutagens. What are mutagens? The agents which cause mutation are called as mutagens. You will get what are mutagens. Mutagens are nothing but the agents which cause mutation. Which agents will cause mutation? Chemical and physical factors like UV radiations. It might be because of error in DNA duplication or replication. These are the mutagens. You will get what is mutagen and give example. Mutagens are nothing but agents which cause mutation, and examples many other. I have mentioned only few, many other. You can refer some other also. Okay, but for an exam point of view, this much is enough. Okay, now we have discussed about what is mutation, what are the causes of mutation, what is mutagen. Okay, now you have here types of mutation. Now this detailed information is not given in your textbook, types and all. It has been mentioned about the further discussion, but not in separate reading. As you know, in NCERT you will get this kind of explanation. You have to search everything and you have to prepare a note. Okay, so because of that reason I am here to prepare a note of it so that you can copy and it will be easy for you all to refer your textbooks. Fine. Types of mutation. There are two types of mutation. One is gene mutation, another one is chromosomal mutation. Okay, these are the two mutations. One is gene mutation, another one is chromosomal mutation. Now here we have gene mutation. Gene mutation means mutation which occurs in gene. Chromosomal mutation means mutation which occurs in chromosomes. Okay, now chromosome itself will consist of a gene. Okay, if there is a change in a gene, we call it gene mutation. If there is a change in number of chromosomes, we call it as chromosomal mutation. Okay, if you find here gene mutation, It's the information you will have. Something like this. Change in the gene. Okay, if there is a change in the gene, it causes gene mutation. It results due to change in the sequence of nucleotides in DNA. If nucleotides have been changed in DNA, then it will result to the gene mutation. Okay, now gene, in this what sequence I have shown, we shall imagine this as a one gene. Okay, this is gene 1. Okay, if this is gene 1, it should have this sequence, but it has been changed. Okay, when it has been changed on a gene, we call it as gene mutation. As I am saying, further there will be a nucleotides which I have not mentioned. This is one gene. One gene can have only 2 nucleotides. It can have many nucleotides. It can have millions of nucleotides together to form one gene. Okay, but I have made you to show this is one gene, very easy because I have used it. Okay, so change in the gene, it results due to change in the sequence of nucleotides. If nucleotide sequence have been changed in DNA, then definitely gene will get changed. So for that we call it as gene mutation. Now in this gene mutation you have point mutation. 2 types. One is point mutation, another one is frame shift mutation. So what is this point mutation? Point mutation is nothing but, okay, it is nothing but change in the single base pair of DNA. If only one base pair is getting changed. Okay, now how many base pairs have been changed here? Here only one base pair has been changed. One base pair. Base pair. We are writing it like this. This is base pair. Base pairs. As you know, base pair is singular, that which means it refers only one pair of nucleotide. Base pairs means it refers many number of nucleotides. Okay, so in point mutation, there is a change in the single base pair of DNA. Okay, only one base pair of DNA has been changed. For that we call it as point mutation. Okay, so what example I gave here, whether it comes under point mutation? Yes, think about it and let me know. Then example, sickle cell anemia and thalassemia. Okay, these are the genetic disorders. Both are the genetic disorders which are caused due to, okay, due to mutation. Because there is a change of only one base pair, that's why it is called as point mutation. If there are changes in many base pairs, we call it as frame shift mutation. Can you find this frame shift mutation? Okay, frame shift mutation. In this what happens, there is a deletions and insertions of base pairs. Okay, there can be deletions, these base pairs which we have given. These are the base pairs. There can be a deletion of base pairs. Okay, deletion of base pairs or insertion of base pairs. Like if I am showing that one, A, T, G, C, 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 G, A. Okay, this is the sequence, right? So its complementary will be T, A, C, G, 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 C, T. So this is the sequence. Now, if in what we said for point mutation, if only one base pair gets changed, we call it as point mutation. If there are many changes, many base pairs are getting changed, we call it as frame shift mutation. That might be because of deletion. It can delete this base pair or it can add one more base pair. Okay, now if this sequence is there, with that in between somewhere here, it has been added. What has been added here? A, and here it will be T. This addition or deletion of many base pairs, not only one, many. It can be 2, it can be 3, it can be not one. If it is one change, then it will be point. If there is more than 2 changes, then we call it as frame shift mutation. Okay, so that frame shift mutation, it can occur because of deletions or insertions of base pairs of DNA. Okay, example you have cystic fibrosis and Tay-Sachs disease. Now, cystic fibrosis is what? Cystic fibrosis is nothing but it is a genetic disorder. As we are saying, it is caused due to mutation. It is a genetic disorder. It is going to affect the those kind of cells which will produce mucus, which will produce sweat, okay, which will produce some wax on our body. Those cells will be getting affected because of frame shift mutation. For that we call it cystic fibrosis. And we have Tay-Sachs disease. This Tay-Sachs disease, it is found in children which are born normal, but they will develop several brain and spinal cord damage. Okay, Tay-Sachs disease you will find in children. They will be normal, but they will start to develop some severe brain and spinal cord damage. Okay, and there are later it forms some error in the DNA, and fat metabolism is affected. Later it will get paralyzed and it will die. Okay, nearly in 3 to 4 years, so it will die. So that is Tay-Sachs disease and some information about cystic fibrosis. Because this information is not there in your textbook, that's why I have talked about it. I did talk about sickle cell anemia and thalassemia. Is it? Because sickle cell anemia and thalassemia, you have a separate topic in detail in the same chapter. I shall discuss about this one in detail there. Which means now we have given sickle cell anemia and thalassemia example of a point mutation, which means what? Only one base pair might have been changed in those two diseases. So that's why it comes under point. Cystic fibrosis and Tay-Sachs, here what you will say, many changes, many base pairs have been changed. It might have been deleted, it might have been inserted. That results in frame shift mutation. Okay, so what we have discussed about gene mutation with the two types, point mutation and frame shift mutation. Now what has been left? Second one, chromosomal mutation. Now for this chromosomal mutation, you have here, there is any change in the number or structure of chromosomes. Okay, if there is any change in the number or structure of chromosomes, we call it as chromosomal mutation. So structure of chromosome, how you will get? You are going to show chromosome something like this. Okay, this is a chromosome. Okay, number of chromosome, how many we have? We have 46 chromosomes, or we can say we have 23 pairs of chromosomes. Is it? 23 pairs of chromosomes we have. Here, if the number of chromosome has been changed, how? There is a 46, it can be, if it has become 47, if it has become 45, if number has been increased or decreased, then it is chromosomal mutation. Okay, so because of the number of chromosomes it can occur, or because of change in the structure of chromosome. Like this is the shape of the chromosome. Then if it is having this kind of structure, okay, this is what again, chromosomal mutation. What has been deleted? There is a deletion of one arm of chromosome. One arm of the chromosome is absent. Okay, either it might be like this. Even the other chromosome, it appears something like this. Here what you will find, both the chromatids are of same size. Here you can find one is long, another one is short. This is also deletion. This part has been deleted. Okay, because this part has been deleted, again lot of information will not be present. If there is a change in the number or a structure of a chromosome, results in what? Chromosomal mutation. Okay, any change in the number or structure of chromosome are called as chromosomal mutations. Hope you understood what does it talk about. Okay, so these chromosomal mutations might be, they may be due to structural change. As I have said, change in the structure. I have deleted here. It might be because of insertion. Okay, some part might have been deleted. Whatever reason it might be. So it might be due to structural change during prophase 1 of meiosis 1. Okay, chromosome structure has been changed. When it can change? It can change in prophase 1 of meiosis 1. Hope you remember prophase 1 of meiosis 1. Meiosis, it is having two types of meiosis. One is meiosis 1, another one is meiosis 2. Is it? In meiosis 1, prophase 1 is the longest phase. In that you have leptotene, zygotene, pachytene, diplotin, diakinesis. Is it? So these are the stages. So if you remember all those, then it will helpful, and there what changes will occur. If structural change can occur in this stage, then it can result in chromosomal mutation. Okay, this is the reason how it might cause. We have two types of chromosomal mutation. One is deletion, another one is duplication or insertion. The name itself shows. Deletion means a part of a chromosome is lost or broken. Okay, you can refer this, right? A part of a chromosome, this part has been lost. Chromosome is deleted, lost or broken. For that we call it as deletion, right? Not only that, here you have duplication or insertion. A particular part of a chromosome is repeated. A particular part of a chromosome, it has been repeated. Now you can find the same chromosome, same type. Now what type of chromosome we have here? Similar to that, one more chromosome is present. In place of 46, if we have 47. Okay, in place of 46, if we have 45, these are chromosome. 46, if there is a 47, then it is duplication or insertion. In place of 46, if it is 45, it will be a deletion. Okay, it might be a part, it has been lost, that will be a deletion. if a chromosome or a part of a chromosome it has been added. Now this is the part of a chromosome which should be present. Now if we imagine this is a normal chromosome, it is present, it is having extra one more copy of the same part of the chromosome, that is also insertion or duplication. Okay, so these are the chromosomal mutations, deletion and duplication or insertion. So it is very easy. It has been asked till now, it has been asked what information in your previous question paper. What is mutation? What are mutagens? Okay. What is point mutation? Give example. What is frame shift mutation? Give example. Only this it has been asked till now. Okay, very important, very easiest. And if you understand this concept, further in future it will be helpful for, because the next chapter which is coming across molecular basis of inheritance, it is very tricky and very easy and interesting. Understanding point is different, it depends on the individual, because I have seen that many students will say that these chapters are very tough and unable to understand. But once if you understand, you can grab it, and easily you can score more, because both the chapters are very important and carrying 10 marks each. Fine. So this is about mutation. Hope you have understood the concept, right? If there are any doubts, any queries, please let me know, and I shall try to change it. And I am trying to upload every day videos, so please be alert. So whenever it has been uploaded, you can see that. From now onwards I wanted to do, inshallah, if in future if it is possible. Anyhow, one request is that if you like my video, please share this link with some of the other PU students so that they also can get benefited. Okay guys, if you have any doubts, any queries, please comment in the comment box. Do like it, and many of them are not subscribing, please subscribe the channel. And we shall meet in our next session with the pedigree analysis. Stay safe, stay healthy.